This video is going to review the lecture for section 8.1 titled Mendel's Experiments. The essential question, what did Mendel's experiments with pea plants reveal that were significant to our current understanding of genetics? There are these 5 learning targets. The first one, who was Gregor Mendel and what was his contribution to genetics? So this is the only known photograph we have of Gregor Mendel, and he is known to be the father of modern genetics. Genetics is the study of heredity, and heredity is our inheritance. So we have an understanding of heredity that we resemble our parents, but genetics is the scientific study of that. And Mendel lived in 1822, was born in 1822, died in 1884, and he was born into very poor circumstances, and his opportunity for education was to become a monk. And so he did, and he entered education as a monk and became a professor as a monk. I mean, he did a lot of experiments with pea plants, published his results, and was largely ignored. To understand this, you have to really think about the time in which Mendel lived. During his lifetime, DNA had not yet been identified as the genetic material of life. I'm not sure if DNA had been identified, but it wasn't known to be the genetic material. So there wasn't an understanding of DNA or chromosomes, or that we had 2 copies of each. None of that was understood. And the current understanding of inheritance was kind of a blended idea that parents contributed genetic material and it just kind of got mixed up, and some of that became you. So a tall parent and a short parent would have an average size person, or a parent with dark hair and another parent with light hair would have a child with kind of medium colored hair. That was just a mixing. That was the current understanding. So why did Mendel choose pea plants, and how did he control pollination? So Mendel picked pea plants. They are easy to grow. They reproduce every year. They're easy to count. But more important was that they had discontinuous traits, meaning that the flowers were either purple or white. The plants were either tall or short. There weren't any mediums in any of these things. And that's how the genetics, these 7 traits on these pea plants work. They're discontinuous. It's either one or the other. We know this to be dominant and recessive, and you may have learned that already. But Mendel tracked and did some pretty solid science with these traits. So there's quite a bit of vocabulary, some of which you may have already learned. Pollination is when pollen lands, the male part, pollen lands on the female part of a flower. And plants can self-pollinate since they don't move and they rely on wind. Sometimes they don't get pollinated by another plant and they have to self-pollinate. Cross-pollinating is when they get pollinated from a different plant. Fertilization is slightly different. We learned this last chapter, but it's when the egg and the sperm join. A trait is a specific characteristic. This is not a new term. So flower color is a trait. Hybrid means we're crossing parents that have different characteristics. So we're crossing a pea plant that makes purple flowers with a pea plant that makes white flowers. That's a hybrid. Mendel controlled pollination. Right? So in this image here, you can see that this is the female part of the plant, this right here. And this is the male part. And the anther here releases the pollen and it lands right on top of the stigma there. That would be self-pollination. So what Mendel did was he took one flower and removed the male parts. Okay, here it is with no male parts. Took a paintbrush and took pollen from a different flower and then transferred it. That meant Mendel knew exactly who the parents were, and he could do have some hybridization where he was in control of who the parents were. So what was Mendel's first experiment, and what were the results? And F1 and F2 are some vocabulary terms we'll pull in here now. So this is Mendel's first experiment. This parental generation here, the pea is parental. They are pure breeding, meaning that this yellow pea parent plant only produced yellow peas, and the green pea parent plant only produced green peas. They were pure breeding. And he crossed those together. That's what this symbol means. We crossed these two parents. And filial is offspring. So the first set of offspring, the F1 generation, turned out to be all yellow, 100% yellow. Then he allowed two of these to self-pollinate, and then they self-fertilized. And the second generation, and the F2 generation, ended up providing lots of yellow peas and a few green. So Mendel counted this many peas, 8,000 peas, which turns out to be about a 3-to-1 ratio, 3 yellow to 1 green. Right? So Mendel did this experiment with all 7 traits, found pure breeding plants for each trait and crossed them. And the F1 generation was all the dominant trait, and then in the F2 generation, the recessive trait showed up again. So here's some vocab: dominant, recessive, gene, and allele. Dominant alleles mean that they are always expressed, and yellow peas is an example. Recessive allele means it's only expressed when there's no dominant option present. So to be green, there has to be no yellow allele present. And then gene and allele are 2 vocabulary terms that we learned last unit. A gene is a section of DNA that codes for a trait, like pea color, and an allele is an option for a gene, like the green allele or the yellow allele for pea color. So if we look at this again, we can see that in this F1 generation here, they're all yellow because yellow is dominant. We see that the green has showed up again, but not very much, 25% or a 3-to-1 ratio. And so the green is the recessive allele. So remember, Mendel didn't know about chromosomes. He didn't know anything about that at this time. These are the results of Mendel's crosses for his first experiment. And so for seed shape, he crossed round and wrinkled, and in the F1 generation got all round. So you can see that in these results, the F1, there is no variation. The dominant is all that's visible, and that was very consistent in his F1 generations. So we're now going to start working on reindeer genetics, and section 1 doesn't introduce some of these concepts, but I think you're probably ready. So I'm going to give just a little bit of background from 8.2 so you can do reindeer genetics. And these are some vocabulary terms that you're going to need to know. Phenotype, which popped up in Crazy Creatures, is what it looks like, and genotype is the genetic makeup. And we're going to start using letters to represent. And you saw this in Crazy Creatures. So if the genotype is homozygous, they are the same. So capital G, or lowercase g, lowercase g. You could probably need to say homozygous dominant if you want to mean G, capital G, and you probably want to say homozygous recessive if you want to refer to little g. You can cheat a little bit and just say heterozygous. That means different, and now we know you have one of each allele, a dominant and a recessive. So let's just take, for example, this experiment here on flower color that Mendel did, okay, where he crossed the purple and the white. Okay? And we're going to use a Punnett square to figure out what's happening. So we know that we have a purple crossed with a white parent. And in Mendel's parental generation, they were pure breeding. And it's kind of typical to use the first letter of the dominant phenotype, which would be purple. So we're going to say this pure breeding was big P, and this pure breeding white was homozygous recessive. P's are a little tricky because they look the same when they're capital and lowercase. So I will make my capital P's really big and my lowercase p's really small. A Punnett square is designed to help you figure out the potential offspring. This is not a guarantee. These are chances. So one parent goes on one side of the Punnett square and the other parent goes on the other side. So this purple parent has two capital P's, and they're either going to give one capital P or the other capital P. They can't give two. You remember in meiosis, parents only give one set of chromosomes. So this is where that comes into play. And then the white parent gives a little p. Now we need to fill out this box with the odds. So this capital P and this little p make a big P and a little p. And then this capital P here and this little p here, and you have the same thing. And pretty quickly you'll figure out that all of the offspring will get one capital P from their purple parent and one lowercase p from the white parent, and they will all be heterozygous, but they will all look purple. So sometimes we write this as 100% purple. Okay? That's how you do a Punnett square. Now, if we look here and we go back and let's use Y for yellow, this parent is capital Y, and this parent is little y. All of these offspring get one capital Y from their yellow parent and one lowercase y from their green parent. Now, if we were to do this Punnett square, we have, remember, they self-fertilize here. So it's heterozygous crossed with heterozygous. And so we have a capital Y and a little y, a capital Y and a little y. So now, these three offspring will be yellow because they have a capital Y. And this one has two little y's, and so they'll be green. And that's where we get this 3-to-1 ratio right here. Okay, so here's a review of those learning targets. Make sure you can answer that essential question, and hopefully that's helpful.